This is Kaylee Robinson. Today I'll discuss Sonic the Hedgehog protein, its associated disease, its primary function in the body, its structure, its pathway, and one of its inhibitors. Today we'll be discussing the disease known as non-syndromic holoprosencephaly. This is characterized by incomplete separation of brain hemispheres that leads to malformations of the face, such as the eyes, nose, and lips. This disease has many causes, but mutations to Sonic the Hedgehog protein is the most common. A specific mutation to the protein is known as P950X, which shortens the amino domain, changing its shape, and the amino domain is necessary for the protein to function. And we have the mutation PC102R, which is also located and affects the amino region. The amino terminus of this protein is where the protein signals from. So deformations in this protein's amino terminus changes the way the pathway is carried out in early embryonic development, which can result in this disease. Here is a table that shows the various different mutations that can cause non-syndromic holoprosencephaly. There are various chromosomal mutations such as the deletion of 13q and 18p. But the most prominent mutation shown that can cause this disease is mutations within Sonic the Hedgehog, which accounts for 5 to 6% of all cases. This protein's main purpose is to regulate embryonic development, which means it helps us stay symmetrical, giving us 2 eyes, 2 arms, 2 legs, really important, 2 hemispheres of our brain, and much, much more. When we're adults, its function is involved in tissue homeostasis, regeneration, and neoplasia. Side note, neoplasia is the uncontrolled growth of cells. So, Sonic the Hedgehog is also involved in cancer development and maintenance as well. All of these functions are regulated by the pathway, which we'll go more in depth into later. In this picture, it demonstrates the localization of the Sonic the Hedgehog protein in a chick's wing bud and the growth of a chick's wing. The darker spots in A, B, D, and E show the localization of the Sonic the Hedgehog protein, and C and F show the bone formation. This picture helps demonstrate the use of Sonic the Hedgehog protein in limb development. We know what this protein's purpose is, but what does it look like? Where does it come from? Sonic the Hedgehog is initially secreted as a precursor protein, which is similar to a zymogen. But zymogen is an inactive enzyme that is cut by another enzyme to turn it into its active form. Sonic the Hedgehog is not an enzyme. Rather, it's a signaling protein, a ligand. So Sonic the Hedgehog's protein, it's translated as this large protein that starts out as 439 amino acids. Its amino terminus is nicknamed Hedge, while the carboxyl terminus is nicknamed Hedgehog. The Hedge regions is mainly the signaling domain, while Hog has autocatalytic properties. After being translated, it's ushered into the ER, where it has cholesterol added to the carboxyl terminus Hog via cholesterol transferase, and then palmitic acid added to the amino terminus Hedge via the enzyme skinny hedgehog acetyltransferase. It is also shortened by various enzymes in the ER to get it into its active form, which is a total of 169 amino acids long. So its active form has 169 amino acids, palmitic acids, and cholesterol attached to it. This next picture is of the ribbon structure of the Sonic the Hedgehog protein. It doesn't show the cholesterol and palmitic acid attached to it, but it does show that it has 2 alpha helices, 6-stranded anti-parallel beta sheets, 2 parallel beta sheets, and 2 calciums and 1 zinc. Calcium and zinc are important for the binding of this protein. Now that we have a good understanding of what this protein looks like and its function, let's talk about the pathway that controls this function. As mentioned previously, Sonic the Hedgehog protein is a ligand, a signaling protein. So the first step in this pathway is for Sonic the Hedgehog to bind to its receptor, PTCH1, on the extracellular side of the cell. This binding disrupts the inhibition of cytoplasmic smoothend, which allows the smoothend to enter the cell to activate SUFU. Activation of SUFU makes the GLI 1 through 3 proteins enter the nucleus where they can coordinate the expression of genes. That is how the protein Sonic the Hedgehog controls embryonic development, such as the symmetry of our limbs, eyes, and our brain. With any good signaling pathway, there are inhibitors. For Sonic the Hedgehog protein, there are a few inhibitors, but we'll focus on the main one, robotnikinin, conveniently named after the villain Dr. Ivo Robotnik, aka Dr. Eggman and Sonic. What this inhibitor does is binds to the Sonic the Hedgehog ligand, which activates the Hedgehog pathway by binding to PTCH1, that receptor that was just previously mentioned. In the absence of repression on smooth end, expression of the GLI3 transcription factors can occur, meaning no expression of the Sonic the Hedgehog protein, which inhibits the pathway. So, in conclusion, this video discussed non-syndromic holoprosencephaly, a condition where the brain hemispheres do not separate, causing deformities in the body due to mutations of the amino terminus of the Sonic the Hedgehog. We also talked about the embryonic development, which helps keep us symmetrical. We talked about how the protein is produced in an inactive form, and then it goes to the ER, where palmitic acid and cholesterol are added to it, and it's also shortened. We talked about the pathway of how it's expressed, and we finally talked about an inhibitor of that pathway, robotnikinin. Now, here are my references for my academic sources. Next, these are my picture references that weren't included in the academic references. This is like the picture of Sonic and Dr. Eggman. I hope you have such a nice day, and thank you so much for listening. Bye!